So now let's start with the balance sheet of aerobic respiration. Okay, so when one glucose molecule undergoes breakdown through aerobic respiration, there is release of heat energy, and some amount of that heat energy is trapped in the form of ATP. That is, net gain of ATP is 38 molecules of ATP. So this net gain of ATP which is released from one molecule of glucose can be calculated on the basis of some assumptions. So now let's see those assumptions. So first assumption is all these pathways are occurring in an order. So orderly, so sequential and orderly functioning of this pathways is taking place. Orderly functioning of this pathways is taking place. So here, during aerobic respiration, what are the pathways? The pathways involved in aerobic respiration are glycolysis, link reaction, Krebs cycle. So all these pathways are taking place in an order, sequential manner, one after the other. That means after glycolysis, it is followed by link reaction is followed by Krebs cycle, and Krebs cycle is followed by ETS. So all these pathways of aerobic respiration are occurring in sequential and order and in an orderly manner. Next assumption is the NADH which is released into cytoplasm during glycolysis. Actually, we know glycolysis occurs in cytoplasm. Site of glycolysis is cytoplasm. So the NADH which is released during glycolysis into cytoplasm enters into mitochondrial matrix. It enters into mitochondrial matrix where it undergoes breakdown through ETS for the formation of ATP. Mitochondrial matrix where it undergoes breakdown through ETS which results in the formation of ATP. So this is second assumption. So we are assuming that the NADH which is released into cytoplasm during glycolysis is entering into mitochondrial matrix, and inside the mitochondrial matrix it undergoes breakdown through ETS for the release of or for the formation of ATP. So that is the second assumption. Third assumption is no intermediate from this pathway is withdrawn for the biosynthesis. So no intermediate from these pathways is withdrawn for the biosynthesis of other biochemicals. That means intermediates which are formed during this pathway are not withdrawn for the biosynthesis of other types of biochemicals. That means they are not withdrawn and they are not used as precursors for the synthesis of biochemicals required by the cell. Next assumption is, so respiration. During this respiration, only glucose becomes the respiratory substrate. So we are studying this respiration process with the glucose as respiratory substrate. So only glucose is becoming the respiratory substrate. So based on these assumptions, we can calculate the number of ATP which are formed by the breakdown of one molecule of glucose through aerobic respiration. So now let's calculate this. Actually, in reality, these assumptions are not correct in reality, because in living systems, these pathways like glycolysis, link reaction and Krebs cycle are not occurring in sequential manner. They can occur simultaneously inside the same cell. Okay, and no intermediates are withdrawn. Actually, the intermediates which are formed during this respiratory pathway can be withdrawn from the respiratory pathway and they can be utilized as precursors for the synthesis of other biochemicals required by the cell as per the requirement of the cell. So during respiration, along with glucose, other respiratory substrates can also undergo breakdown. That is proteins, fats, organic acids, they can also undergo breakdown. So all these assumptions are not really correct. In reality, they are not correct, but the calculations which are based on these assumptions are only of theoretical value. Now, let's calculate the number of ATP produced by one molecule of glucose when it undergoes breakdown through aerobic respiration based on these assumptions. Okay, so let's see, based on these assumptions, how many ATPs are formed by the breakdown of one molecule of one molecule of glucose through aerobic respiration. So stages during which this ATP is formed, and then source can be either SLP or OP. And number of ATP. Okay, first stage is glycolysis. So during this glycolysis, what happens? ATP can be formed by SLP, that is direct formation of ATP. Through SLP, how many ATPs will be formed? 4 ATPs will be formed. That is at 7th step and 10th step. At 7th step 2 ATPs and at 10th step 2 ATP will be formed. Among these 4 ATP, 2 ATPs are consumed. So what is the net gain of ATP? 2 is the net gain of ATP. So 2 ATP is the net gain. Next, coming to OP, oxidative phosphorylation. Oxidative phosphorylation involves the synthesis of either NADH2 or FADH2. During glycolysis, what is synthesized? NADH2 is synthesized. How many molecules of NADH2 is synthesized? 2 molecules of NADH2 is synthesized. So 1 molecule of NADH is equals to how many ATPs? 3 ATPs. So 2 molecules of NADH2 into 3 ATPs. So totally how many ATPs? 6 molecules of ATP. Right. Next, coming to Krebs link reaction. Second step of aerobic respiration, that is link reaction. So during link reaction, ATP formation through SLP is absent. There is no direct formation of ATP. There is formation of ATP only through OP. So how many NADH2s are formed here? 2 NADH2s. So 2 NADH into 3 ATP. Again 6 ATP during link reaction. Next, coming to Krebs cycle. Third step of aerobic respiration, that is Krebs cycle. So during Krebs cycle, ATP can be formed through SLP and even through OP. Oxidative phosphorylation. Through SLP, how many ATPs will be formed? Actually, 1 ATP will be formed per 1 cycle of Krebs cycle. But for 1 molecule of glucose, we have to take 2 cycles of Krebs cycle, because when 1 glucose molecule undergoes breakdown, enters into aerobic respiration, by the end of glycolysis, it undergoes partial oxidation into 2 molecules of pyruvic acid. Those 2 molecules of pyruvic acid enters into mitochondrial matrix, which enters into link reaction or oxidative phosphorylation, by the end of which 2 molecules of acetyl-CoA will be formed. So per 1 acetyl-CoA, 1 cycle of Krebs cycle occurs. So per 2 molecules of acetyl coenzyme A, 2 cycles of Krebs cycle occurs. So per 1 cycle through SLP, 1 ATP will be formed. So per 2 cycles of Krebs cycle, how many ATPs? 2 ATPs will be formed. 2 ATPs. Next, coming to OP. So during this Krebs cycle, through oxidative phosphorylation, both NADH2 and FADH2 synthesis occurs. So how many NADH2s will be formed per 1 cycle? 3 NADH2s. So per 2 cycles, 6 NADH2s. So 6 NADH2, 6 NADH into how many ATPs? 3 ATPs. So 6 3s are 18. So 18 ATP. Next, FADH2. How many FADH2 actually per one cycle? One FADH2. Per two cycles of Krebs cycle, 2 FADH2. So, 2 FADH into 2. Per one FADH2, how many ATPs will be formed? 2 ATPs by its breakdown through ETS. So, totally how many ATPs? 4 ATPs. So, total number of ATPs are, total number of, in glycolysis 8. Then during link reaction 6, so 8 plus 6, 4, and then during Krebs cycle, 8 plus 6, 14, sorry, and during Krebs cycle how many? 24. So totally how many ATPs are formed? Through Krebs cycle 24, here 6, here 8. So 8 plus 6 plus 24. So total number of ATP formed is 38, which is the net gain of ATP. So total number of ATP means 40 if we include these two ATPs also of glycolysis, and net gain of ATP means 38 because among 40 ATPs, 2 ATPs are utilized, where that is in the first step of glycolysis and third step of glycolysis. So net gain of ATP is 38. So this is all about the balance sheet of aerobic respiration. And in the next video, I will be starting with the efficiency of aerobic respiration. Hope you understood all these concepts. Thank you, students.